guess going back to your specific research on the system, so you're looking at the export apparatus, is that right?
We are looking at all the aspects of how this machine works, as you can imagine. So we are looking at aspects of not just the, for example, we would like to know the atomic composition of the machine. We are making inroads into essentially getting an atomic map of the entire machine, so that we will know where each atom of each protein will be located. We are trying to understand the actual mechanism by which proteins go through a central channel of this machine, and the whole energetics of how those proteins progress in their trip, so to speak, from the inside of the bacteria all the way to mammalian cells. So there are still many things we do not understand. That's absolutely for sure. And as far as this sorting platform, I mean, obviously this is sort of the initial salvo in an area that undoubtedly is going to require more effort from our part to truly understand how this platform sorts the different proteins and then gives them to the actual delivery machine so that the machine can deliver them, and so on and so forth. So.
And the proteins in the sorting platform, you already knew that they were part of the system?
Absolutely. We knew that these, we bumped into this a little bit by accident. We knew that they were a subset of the proteins that make this type 3 secretion system, if you will. That is, proteins that are absolutely necessary for the workings of the machine, that if you remove them, the machine doesn't work. And these were a subset of proteins that we knew were inside the bacteria, but we knew they were essential because if we remove the gene that encodes them, the machine would not work. That we know for a while, but we in essence were trying to figure out what were they doing, and then is when we bump into this sorting platform, because the idea of the necessity of an order, it was before this work probably was a bit more of a concept than this added more evidence to the concept that indeed you need to have an order. There was some evidence out there, but probably not enough to convince everybody. I think now we have provided more evidence even for that aspect, but I think this is more or less well accepted in the field that you need an order. But the main focus is not just to really prove that there was an order, but rather how that sorting takes place. What we are trying to do at the moment is to actually, we are attempting to actually see the sorting platform, because we know that is in relative terms is a fairly large structure. So we know how to isolate it, and we are trying to utilize techniques such as electron microscopy and things of that sort to visualize it and to be able to define its structure. And then, of course, layer on that will be how proteins are engaged by this platform, how are they recruited. We know a little bit about it, but certainly we have quite a ways to go to truly understand how it works.
The chaperones bring the effector proteins. Do they also bring the
translocases?
Yeah, translocases.
Yes, they do. They do. Oh, I mean, they're
it's
different.
All in order.
Correct. Okay. Yeah, because you are referring to some proteins that are also coming through this needle complex, as we call the machine that is this nanomachine. There are certain proteins that need to make a little pore on the mammalian cell membrane. Okay. And those necessarily have to go there first, because if you don't have the gate to get into the mammalian cell, there is no point in bringing the actual proteins that are going to be doing stuff inside the cell. So that's basically one of the sort of key elements of this hierarchy. That first you got to assemble that pore, and those in our paper we show that those have priority sorting platform. They have more priority than, say, the what we call effector proteins. That is, the proteins that are going to be carrying out some chore inside the mammalian cell.
Right. And also, the secretion system is also used by other bacteria, is that correct?
Right. So that's another exciting part of this, is that, as you could probably imagine, something as remarkable as this organelle, and it's so successful at doing what it does, which is to be able to deliver proteins into mammalian cells. But more than that, I mean, bacteria have figured out how to deliver bacterial proteins into cells in a much simpler manner. For example, regular toxins, which are proteins that bacteria make that sort of do nasty things to cells, but that's a much simpler kind of mechanism. But that mechanism simply involves delivering, one or two activities into the cell, and bacteria usually kind of put them out, and then somehow these toxins get to their targets. This is much more complicated. Probably these machines evolve out of the need to deliver many activities to the same cell. That is, if the cellular processes that bacteria needs to modulate in the target cell are complex, maybe 1, 2, 3, or even 10 activities cannot get the job done. So the question is, how do you deal with that kind of problem? So these machines are ideal because they deliver many proteins, in the case of Salmonella, upwards of 60, to the same cell in a coordinated fashion, say in time and space. So that kind of complexity cannot be accommodated just by simply throwing proteins into the outside of the bacteria and hoping that they will hit the cell that they need to hit. So these machines are very good at that, and consequently you could predict, obviously, that it would not be an exclusive domain of Salmonella to have such a remarkable successful device. Evolution, when evolution comes up with something successful, it tends to be spread in nature. So many pathogens do use versions of these kinds of machines, many important bacterial pathogens. Interestingly, also symbionts and pathogens not just of mammals, but also plants. So this is a remarkably successful piece of evolution, if you will, that has been put to use in the context of different types of bacteria interactions with some kind of, eukaryotic or nucleated entity, from a plant to, amoebae to humans. So the consequence of that is that you can imagine that if one comes up with a drug to interfere with this machine, doesn't necessarily need to be a sort of a Salmonella exclusive drug, although you could probably engineer something like that because they're slightly different one from the other. But in reality, you could imagine designing something that would be good for a family of pathogens, for example, those that cause plague, whooping cough, dysentery, many important pathogens that use devices like this to be able to cause disease.
What are the first next steps that you're taking in your research, I guess?
Well, there are many next steps that we're taking, but on this particular story that we were discussing, I think we're now really focused on trying to visualize this platform and assembling it with spare parts in a test tube. So sometimes to really understand mechanisms, you need to reduce complexity to some degree of simplicity. So we are trying to build this platform from the components from the ground up and see if we can sort of get it to work in a test tube, so to speak, so that we can truly understand how it works.